Hello, I am Mr. Wilson from CSEC Biology, the cover page. If you will be doing a biology or human and social biology exam and genetics is your concern, worry no more. Stay with me as I work out and explain Punnett square, genetic cross, and sex-linked inheritance. What is sex-linked inheritance? It refers to a condition or genes that is carried on the sex chromosome. In males, the sex chromosomes are X and Y, while in females, the sex chromosomes are XX. It is always possible for both male and female to inherit the X-linked trait, as they both have X chromosomes. Y-linked traits are, however, confined to males since they are the sole carrier of Y chromosomes. Recessive traits that are associated with the X chromosomes only affect female if it is inherited in the homozygous recessive form or genotype. If a trait for the recessive allele is found only on one of the X chromosomes in women, they are not affected by the trait, as the condition trait is usually masked by the dominant allele. In such case, the woman is called a carrier and is able to pass on the recessive allele to her sons. Even if a father did not have the trait, she will also be able to pass on the trait to half of her girls. Her daughters, however, will also be carriers. The male is not able to inherit two recessive alleles. This is due to the size and type of chromosomes, XY. However, if they inherited one of the recessive allele, they would be affected by the trait, as the X chromosome is taller than the Y chromosome. Hence, there will be no dominant allele to mask the recessive trait. Here we have a diagram of the chromosomes, X and Y, male and female, and we're looking at common H here being the recessive allele and capital H being the dominant allele. Now what we're saying, it is on the X chromosome that the trait would be. Now in the case of the male as such, once the male has the recessive allele on the X chromosome, the male will be affected by the trait. On the other hand, the female here has one recessive allele and a dominant allele as well. In this case, the recessive allele is masked by the dominant allele, capital H here. The capital H is the dominant allele. As such, it will mask the common H, which is the recessive allele here, hence making this female a carrier. Now, a carrier female is not adversely affected by the trait. However, she will pass on the trait to her daughters, but her daughters will only be carriers. Hemophilia. This is a disease in which the blood fails to clot. Hence, even with small cuts, the individual bleeds more than normal. Allele capital H is the normal clotting allele or dominant allele, while allele common H is the recessive allele for the trait. Color blindness or red-green color blindness. In this case, individuals suffer from this condition, are not able to see some colors. The condition is observed with red, green, and blue. This can cause major problems with indicating lights, colors, and the perception of fruit maturity. Let's look at an example of the genetic cross or the genetic diagram as we would have it. First, we're defining the allele here. So the first individual is a female and she has the allele for normal blood clotting. The second individual is a carrier female. She has the recessive allele, although it's masked by the dominant allele. And the dominant allele is the capital H for those persons who are just starting genetics. Now, this person will not be adversely affected by hemophilia. However, this individual will be able to pass on the hemophilia to the F1 generation. The third female is a female that is affected by hemophilia. She's a hemophilic, which means that, of course, she is adversely affected by the condition, and so too she'll be able to pass on the condition to her offspring. The other individual is a male, and this is a male for normal blood clotting, as this male has the dominant allele H, which will cause this individual to have normal blood clotting. The last male is a male that suffers from the condition hemophilia, so we say that he's hemophilic, as he has the recessive allele for hemophilia. Now that these are well defined, let us look at doing a genetic cross, which is well animated. At any point you can stop, replay the video to ensure that you have a good grasp of doing the genetic cross. So we start by looking at the question. The question we have here is that a normal male is crossed with a carrier female for hemophilia. We're supposed to determine the phenotypic ratio of the F1 generation. We're supposed to show all steps as we complete the genetic diagrams. So here we go. We know that the genotype for the individual must be written, and we know the phenotype, of course, for the two individuals. One, of course, is a normal male, and the other is a hemophiliac or hemophilic carrier female. So here we have just the male female as phenotype, nothing attached there. However, the female should have been written there as a carrier, female carrier. Now the genotype is well written. It's a normal male, and you're seeing there the dominant allele, capital H on the X for the male, so he's not affected by the disease. However, the female here, which is a carrier, and the phenotype I said before should have been female carrier. However, the genotype is correct. The genotype for the carrier female is a dominant allele, capital H, a recessive allele, common H, and they are both on the X chromosome. Now the next thing we are going to do, we are going to be identifying the gamete. So here we go, the gamete with the allele there. Good. So that is your first step when you get into the exam. You're going to be identifying the phenotype from your question. You're going to be identifying the genotype based on what was given to you, and then you are going to break away with the gamete showing the chromosome and allele, and of course then we're going to be moving into segregation, or what we call random fertilization. Now look what happens here. So we're trying to identify the F1 genotype, so that is the first part of the cross, and here we go with the second. So observe, they are color coded just to show you where the chromosome, the allele, where they are coming from to make the F1 generation or the offspring. Right, there we go. Now please make note of this as we move to the other side. So there we go. Just let me pause and identify this bit. If you are observing, here we have two arrows with two chromosomes. So here this is completed. Here we have two arrows with two chromosomes. Here this is completed, right? We have one here, which means that we need to have another coming from it. We have one here, which means we need to have another coming from it. Same too for this chromosome. Now this here is completed. There's nothing else that can come from this because we are doing monohybrid inheritance. All right, we're just looking at one trait. So here we go. Now completed, and we have just one other. All right, so if you look here, completed, 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 but this here still needs another arrow, and this here still needs another arrow. Here we go. One down. So here it is completed here. So now this needs another arrow, and of course this needs an arrow just the same. All right, so we're going to be crossing from here to here with that arrow. Here we go. All right, that completes the genetic cross, and exactly how you're supposed to be doing it for your CSEC examination. Remember, at the top here, you must be paying attention. At the top here, it should have been female carrier. Female carrier. We only have female there, but it should have been female carrier. Please ensure that you make that correction. Now we're supposed to be identifying the phenotype, but we could identify everything here before. Here we have the phenotype, which is taken from the question, and the genotype. Based on our interpretation and our prior knowledge, we know that this is what it should be. And then we come down to the gametes. Here we have random fertilization here, or some might say segregation. And then we come to the F1 generation, which stands for the first filial generation, the first set of offspring from these parents. Then now we need to identify what these offsprings are. Are they affected? Are they male? Are they female? That type of thing we need to identify. So let's look at the phenotype. So here we go. So the phenotype for the first individual, it's a female normal, which means that she is normal for blood clotting. Her blood will clot perfectly well. Then the other, we have a male, is a hemophilic male, as he has one recessive allele, and of course the only allele that he could have had, since the allele is linked to the X chromosome. Then we look here, we have a normal male, since he has a dominant allele H linked to the X chromosome. Here we go again. And the final, we have a female carrier, and if you remember when we started out, we said this is what it should have been, a female carrier. And a carrier, because it's pretty much in the heterozygous state, as we would have said for the allele, where we have a dominant allele and of course a recessive allele. The dominant allele here being capital H and the recessive allele being common H. Remember, at all times for genetics, we usually use a capital letter to represent the dominant allele and of course a common letter to represent the recessive allele. Now remember, the dominant allele is that allele that will be expressed once the genotype has that allele, then it will be expressed in the phenotype. However, the recessive allele will not be expressed unless it is in its homozygous recessive state, which means that you have too small h, except in the case of sex-linked inheritance with the male. Let's move on and look at the phenotypic ratio for the F1 generation. So ratio is always going to be in its simplest term. So here we go, we have male, female, there we go, and then the ratio 1 to 1 to 1. So what it says here is that we have 1 female normal, 1 male hemophilic, 1 male normal, and of course 1 female carrier. And that's it for the F1 generation. Now we're going to be solving the same problem using the Punnett square. We start by pointing out this bit. You have to pay attention here. So if you copy the slide, you are going to be missing this bit. All right, so here we should have started out with female carrier. So female carrier, because this is what this is saying, that this is a female and she's a carrier for the disease hemophilia. Now this would have been your phenotype at the top here, and then we would have had our genotype, which is usually represented using the letter. Remember, the phenotype is a characteristic that you can see. All right, and we would have done that several times, and we will continue to do so. So here we have our genotype, and we're now breaking away to the gametes, which show the allele. There we go. And this is how you're supposed to be doing it for your examination. You should ensure that you can identify the gamete as they fall from your genotype. Then we go on to the Punnett square. The Punnett square is just a rectangle with two horizontal lines 2 vertical lines, and that creates 9 cells. And then for the first cell, we're going to be splitting the first cell in 2. Then we're going to be identifying, of course, what we have in the header row and header column. So here we would have male, as will be shown with a color coding, and here we're going to be having female. So look what we have there. All right, male. So you would write or use the symbol here. Then for the female, we're going to be having, all right, and you would have seen where we got that bit of information. And the same is true. We would have written here female carrier, and over here we would have written, of course, normal male. So normal male here, that is the parent, the parent genotype, and here we would have had the, of course, female parent genotype. So the male genotype over here, and of course the female genotype here. And now we're going to be populating the cells. So here we go, and you can see the H coming from the white there and black there. So you see where they intercept, and it makes that. And there we have it again, H coming from to your left, and of course the Y coming from above. If you look at your screen, that's what we're saying here. This H here coming from over here, this Y coming from here, and this XH coming from here. And of course, I'm being very, very simple here for those persons who are doing it for the first time, and I want you to get it. But as soon as you are a pro on it, please start using the correct terms. So here we go again. Chromosome and allele coming from over here, and now again that's a female from the female, and now from the male we have the white here. Remember, these colors are just used for you to identify them easily. All right, so in your exam using one color pen, it is fine. All right, so here we go again. Wonderful. And now we have to determine the F1 generation phenotypic ratio, and like we did before, it's going to be pretty much the same. Remember, if you have not followed clearly the procedure in working the Punnett square out or the genetic cross out, please replay the video however many times you wish until you would have mastered the skill. Also remember, we're doing several crosses. So far we have done test cross, we have done inheritance of sex, now we are doing sex link inheritance, and we're going to be doing others. It's very important that you understand. It is very important that you understand the rules that are associated with all these cross. Do not take it for granted that, of course, it's just a cross and we are just crossing. There are rules for all the different crosses that we're doing. Please pay attention to the rules. Now I think you'd love to try some. Here I've given you 4, right? I want you to identify the phenotype of the parents. You remember when we identified our phenotype earlier, we saw that the lady, for the female, we did not completely write the phenotype. So you should write the complete phenotype for your F1 and your parent, and of course you should write the genotype as well. Don't just write male and female. If they suffer from the condition, you need to say whether they are a female carrier or a female that is affected by the condition, or a male normal or a male hemophilic. That type of thing you need to ensure that you have written. Now you're supposed to identify the phenotype and genotype of the F1 generation, and the F1 generation stands for the first filial generation. That's the first set of offspring. Then you are supposed to use both genetic diagrams to show your working, which means that you're going to be using the genetic cross that we have used, and you're going to be using the Punnett square to solve. Both you're going to be using to solve to ensure that you have mastery of both. Then you're going to state the phenotypic ratio for your F1 generation. Tell us what you would have seen based on your cross. All right, and I think that one of these might be similar to the one we've worked out, so probably you want to start from there, where we had a carrier female. I think we had a carrier female that we had crossed with a normal male. I'm not seeing that here. All right, but try that one if you so wish, so that you can, of course, be able to solve them with great ease. Thanks much for watching, and I'd love for you to subscribe to my channel. So just below this red banner, you're going to see something that says the cover page. Please click on the cover page to subscribe. Thanks much for watching.